Welcome back to Unit 3, which is all about genetics, and today we're going to dive into a much deeper look about chromosomes themselves. So let's start with differentiating between prokaryotes and eukaryotes. So in prokaryotic genetics, prokaryotes don't have a nucleus. That's like the main thing about prokaryotes, is that they do not have a nucleus. So genetic material is just kind of like free-flying in the cytoplasm in an area called the nucleoid. Prokaryotes have a single chromosome and circular DNA, and it's considered naked because it doesn't have proteins on it. Some prokaryotes also have an additional circular DNA molecule called a plasmid. You can kind of see that on the bottom. The orange circle is a plasmid, and then the pink circle is their circular DNA. So bacteria cells exchange plasmids via their sex pili in a process called bacterial conjugation. And you can see that in the bottom picture. So you have a donor cell and a recipient cell, and that sex pili in the first picture is like starting to come out and get into the other bacteria. And then in here you have a conjugation bridge forming, and that's just a bridge between the two bacteria. And this horizontal gene transfer happens where you make a duplicate of this plasmid and it goes into the recipient bacteria, and then you have a duplicated plasmid. And this is ideal for gene manipulation because you can just make a ton of copies of the same plasmid and just do whatever experimentation you need to do on it. For eukaryotes, it's a little bit more complicated just because our DNA is wrapped into chromosomes through a number of steps. So we have linear molecules of DNA, and so going from left to right in this picture is how I'm going to be describing it. So you have your DNA, and it's put together and wrapped around 8 histone proteins to form a nucleosome. So that's what you see here. Then these nucleosomes are linked by additional histone proteins, and they form chromatosomes, which you can see here. The genes that are on the nucleosomes and the chromatosomes are your active genes. So even though they're wrapped up really tightly, they're easy to unwrap. And the reason that we care about that is because when we go through DNA replication and transcription and translation, you need to be able to easily access those active genes. Once you get to the chromatosomes, those are wrapped up even more and coiled up to form solenoid structures, and they form a 30 nanometer fiber, which you can see in these next two pictures. These are where the less active genes hang out. Yeah, they're important, but they're not as important as the active genes, so they're a little bit more wrapped up and a little bit more challenging to uncoil because you don't need to uncoil them as frequently. And then this fiber is compressed and folded to form chromatin, and then the chromatin is supercoiled to form chromosomes. So all of that happens so that you can form these tightly coiled chromosomes so that it can all fit in your cells. So general side-by-side comparison, pro versus eu. Prokaryotes is in the cytoplasm, naked, genomes are compact, can contain plasmids, and is circular, whereas eukaryotic is contained in a nucleus, bound to proteins, large amounts of non-coding and repetitive DNA, no plasmids, and is linear. So when we're looking at chromosomes, we really need to understand how you can find genes and kind of how you would write that, what kind of nomenclature you would use. So each chromosome has a constriction point called a centromere. This divides the chromosome into two arms, the P arm and the Q arm. The P is always the shorter arm and the Q the longer arm. So if I were to say 7p12 region of a chromosome, the way that would break down is the first number, 7, is what chromosome you're looking at. P, meaning the arm, it's either going to be P or Q, and then 12, the location of the gene itself. So you're looking at the G-band location, and you can see that up here. So this is chromosome 7. We're looking at the P arm, and then 12 right here is the G-band location that we would be looking at. So we would be looking at the epidermal growth factor receptor. That will come in handy as we continue moving through looking at different chromosomes. So different types of chromosomes. There's metacentric, where the centromere is in the middle, and the P and Q arms are comparable. Then there is the submetacentric, where the centromere is off-center and you have shorter P arms. The acrocentric, where the centromere is really off-center and much shorter P arm. And then the telecentric, where the centromere is found at the end of a chromosome and no P arm exists. This does not happen in humans, but it can happen in other organisms, so I wanted to bring your attention to it. Now, we talked about mutations last time, but these are just kind of the general types of mutations that might pop up in various questions or various assignments that we do. So duplication is when you duplicate a base pair, right? You've duplicated the B. Deletion is when you delete one, so we've deleted the B here. Inversion is when we swap sides of the arm, so we took the ED from here and the BC from here and swapped them. And then translocation is when we are moving things from one area to another. So you can see here we moved this E and F to over here. We swapped these N bits. So that's just changing the location of those base pairs. Moving on to another definition that you may remember from meiosis, homologous chromosomes. So homologous chromosomes is just two copies of a chromosome. So you get a maternal and a paternal copy of a chromosome, and they have the same genetic material on the chromosomes. They have the same structure, size, banding pattern, centromere position. All of that is the same. Those are considered homologous chromosomes. So again, you get one from mom, one from dad, and they have the same genetic material on them. You might have different alleles on them, but they have the same genes and patterns and size. So moving into that even further, let's talk about haploid versus diploid. So a haploid set of chromosomes is one set of chromosomes. You get one copy of a gene for each trait, typically sex cells. It's all our sex cells, but that's typically what you know of as haploid, though not always true. And then present in bacteria and fungi. And then diploid is having the actual pair of homologous chromosomes. So you have two copies of each gene for each trait. Your somatic cells, your body cells, and is present in most animals and plants. So autosomes versus heterosomes. Again, we're just diving even further into all of this. So autosomes are your body chromosomes, like all of your normal chromosomes. In humans, it's 1 through 22. And then your sex chromosomes or heterosomes is your last set of chromosomes, which is set 23 in humans. For females, we have XX, and for males, XY. The Y chromosome contains genes for developing the male sex characteristics. And if you don't get a Y chromosome, then female sex organs will develop. So based on this, and based off of your understanding of how genetics works, the father is always responsible for determining the sex of an offspring, because the father is the only one that can contribute a Y to the offspring. Now that's our sex determination system, but there's others. So let's talk about a few others. So there's the XO system, which is like insects, like grasshoppers and crickets. So females are XX, and then males only have one chromosome, the X0. So generally, sex is determined by the overall level of gene expression. The more gene expression you have, you're considered female. The less gene expression you have, you're considered male. The ZW system is common in birds, reptiles, and some insects. Males are ZZ, females are ZW. And then the haplodiploid system, which is common in certain insects like ants and bees. If you know anything about ants and bees before we even get started reading this, then you know that they have a queen system, and this is how they do it. So unfertilized eggs develop into haploid individuals, which are always male, and fertilized eggs develop into diploid individuals, which are females. However, the only ant or bee that can actually reproduce is the queen. So all of the other females are sterile. So even though you have this differentiation between male and females, only the queen can reproduce. So another piece of information that you've definitely seen before but maybe forgot about is karyotypes and karyograms. So karyotypes are the number and type of chromosomes in a eukaryotic cell. So that's just knowing the number and what kind of chromosome. So like you have 22 autosomes and 1 sex chromosome in a eukaryotic cell of a human. But karyograms, that's like what this picture is. So that's probably what you're more used to. So a karyogram is that visual representation of the chromosomes. All right, now we're going to get into a process called autoradiography. So this was used to measure the length of DNA molecules and provides an actual visualization of the DNA and of the chromosome with the use of an electron microscope. So you can take a minute and pause and check out this picture, but what I really want you to understand is that at the end, you get this autoradiograph of the DNA that you can see, and you can really understand its size in relation to other chromosomes, where the centromere is, the size of the arms, all of that. Now, John Cairns is the person who is responsible for discovering autoradiography. He was the first to measure uncoiled chromosomes, which gave more accurate measurements. So this was like the beginning of understanding that there were different sizes of chromosomes. And he was also able to demonstrate some really big events during replication that we didn't know happened. So like during replication, there's a replication bubble that happens. And that's when you are replicating specific areas of the genome. And that replication is bidirectional, right? You have a leading and a lagging strand. Okay, so for the last bit, it's just like some quick recaps of things maybe you know, but I'm just going to go over again. So for chromosome numbers, organisms with different diploid numbers are unlikely to be able to interbreed, right? You can't interbreed with an organism that has 20 chromosomes with an organism that has 40 chromosomes. You're not going to be able to produce a viable offspring. But if the amount of chromosomes, your diploid numbers are similar, it's not unheard of that you can create an offspring. For example, the horse mating with a donkey can produce a mule, but the mule is infertile, right? It doesn't have a haploid number because it doesn't have an equal, even diploid number, meaning that it cannot produce a viable offspring. Diploid number comparisons, this is similar to what we were talking about at the end of last lecture. So again, you have your chromosome numbers, and you can see that there's no real information that you can gather from this. It's just general information. You can't determine if organisms are more intelligent than others just based off of their diploid number or their genome size. And then last but not least is variation in genome size. So the larger the bar, the more kilobases that you have in their genome, so the larger the genome. Now we're a mammal, so ours is actually quite small in comparison to, say, flowering plants, which is absolutely massive. And the reason that flowering plants is so big is because they can change their genome like all the time, right? We learned about polyploidy. It's this idea of changing their genome and continuing to get larger genomes leads to a larger genome size. That makes sense, hopefully. So again, this is just showing you that there is a huge variation among all organisms in genome size, amount of base pairs, amount of diploid chromosomes, everything. And it does not necessarily lead to any understanding of intelligence in the organisms themselves. All right, I'll see you next time.